Hi everyone. This week we are talking about Chapter 5, antibodies and their structure and function. Let's get the slideshow up. All right, here we go. So at this point we've talked about several processes. First we spoke about the innate immune system, where we started off with how phagocytes serve as antigen-presenting cells. They encounter a foreign, and we're looking at this over here, they encounter a foreign pathogen or particle called an antigen, something foreign to us. The cell then uses some proteasomes, or just little packages of enzymes to digest the antigen, and then breaks that antigen into little small pieces. And we remember that the antigen transporters within the cell, they're called transporters associated with antigen processing, TAP1 and TAP2, then bring the peptides into the endoplasmic reticulum of the antigen presenting cell, and the peptide is then either bound to MHC class 1 for endogenous pathogens like viruses, or MHC2s for exogenous pathogens like bacteria. The antigen then binds at the antigenic cleft, where the MHC then moves from the endoplasmic reticulum through the Golgi complex and is brought to be displayed up on the cell surface, where, depending on the process for class 1 molecules, the antigen gets presented and bound to the T cell receptor of cytotoxic T cells, which then release cytokines in the forms of perforins and granzymes, resulting in the destruction of the target cell. So that's this side here. And if the pathogen is of an exogenous nature, like bacteria, the MHC2 complex binds to the TCR, the T cell receptor here, of a T helper cell that results in a number of things. This virgin or naive T cell is then activated to do a number of things. It'll proliferate and make copies of itself as an activated T helper cell. It will release cytokines to activate B cells to both proliferate and form some memory cells, and it'll transform into plasma cells where antibodies and other active cells induce an inflammatory result. We call this entire process the role of T cells in response to antigen presentation, the cell-mediated immune response. And so I should remind us, we can produce memory T cells, we can activate B cells, and we can activate cytotoxicity, and we can also activate neutrophils and macrophages to induce some inflammation. We also know that B cells can be similarly activated when a mature but unchallenged B cell encounters an antigen in that secondary lymph tissue, the spleen, the lymph nodes, and where B cells become activated after encountering that antigen. Similar outcomes arise in that the B cell is stimulated to transform into a plasma cell to produce antibodies specific to the antigen encountered. There can be some memory that's built here as well, though it's certainly not as effective as the memory that's built around from the T helper cells, but the real star of this process is the production of antibody by B cells. In this case, the B cell didn't require the action of T cells to aid in its proliferation and antibody production, and so we call this process, or these types of antigens, T cell independent antigens, or T independent antigens. And that's the process we see here on the left screen. The biggest issue with this is that the memory is less likely to occur, or even weak at best. So we call the process involving antibody production through B cells, we are going to call this humoral immunity. And I guess additionally, this one last slide right here, one last illustration here, we're kind of reminded of the bridge between the innate and the adaptive. in that the antigen gets processed by T lymphocytes and macrophages in a way that we've discussed previously, and the immunogenic part of the antigen can be presented to the B cells when the B cells have specific receptors that will bind to the same surface of the antigen as the T cell. That kind of bridging, remember we had markers on both B and T cells that will facilitate that, like the CD40 marker. This represents, and then after these bind, we have, it triggers the T cell, releases cytokines, and triggers what? It produces memory cells. These are the really super effective memory cells, and the plasma cells are initiated to produce immunoglobulins. And again, that's kind of the star of the humoral response. And so after this really quick and cursory review, that brings us up to the start of today's lecture about antibodies. I just wanted to remind us of kind of the processes that get us to the point of antibody production. And here we are. Give me 1 second. I just want to, well, I can do it from here. I just wanted to set myself up with the pointer. Sorry about that. Let me just go through. There we go. Just wanted to, I couldn't do this until the slideshow was up and running. There we go. All right. The antigen, so when we're talking about the humoral immunity that we just talked about, it was about antigen production, right? So once that first and second line of defense have been breached and they are not able to eliminate the pathogen, cellular and humoral immunity respond. Cellular, we've seen, initiates the T cells, and humoral immunity is the response mechanism by the production of antibodies. In this case, the antibodies are very, very specific to the foreign material or the antigen that gets that causes the reaction of the humoral immunity. We're going to see that in just a second, how the response is very, very specific. I'm not going to spend time here because we know, but antigens and immunogens, remember, immunogens can elicit immune response, antigens bind to the product of the immune response. Antigens are foreign. They're foreign particles or molecules, bacteria, funguses, yeast, pollen, allergens, and anything that's foreign to the host. And antigens, small pieces of those antigens, after they're processed and presented, we will present a very small piece, a peptide of that antigen in the presentation within the immune system, and we call those the antigen determinants or epitopes. Those are the really small pieces of the antigen or the molecule that is termed antigenic. Remember, you can have a large molecule like a full bacteria that has multiple different epitopes on its surface that are identified as foreign. You could have a very, very small particle called a hapten. It's very small, and it may have only one epitope because it's such a small molecule. So epitopes don't have to be numerous in number, but if they aren't, like in the case of a hapten, the size and the number of epitopes is really going to determine how antigenic that molecule is. So let's get to antibodies. Just like antigens are the initiators of the immune response, antibodies are the main response of the humoral immune system. We're going to talk more about specific antibodies in detail shortly, but I wanted to address their function at this point. Another word for antibody is, of course, immunoglobulins, and there are 5 major classes of immunoglobulins: IgA, IgD, IgE, IgG, and IgM. IgM is going to be our first responder. We're going to see this one first, and IgG is going to be our most prolific responder. Most of our antibodies are IgG in nature. Immunoglobulins, we saw in a prior lecture briefly that it's illustrated for us in the form of a Y. And there are two types of chains that create the structure of an antibody. There are these two heavy chains. These are the long chains here. Those are called heavy chains. And then we have the light chains, these smaller ones. So there's two heavy chains and two light chains in each antibody structure. Within the antibody structure, it's not shown here, but there are disulfide bridges that are going to hold this together. And there's a little hinge here. We're going to look at that in just a few minutes. The heavy chains, we also call them H chains. You'll see them referred to as H chains. The heavy chains have a variable region, a small variable region here, and the rest of this is called the constant region. Similarly, light chains have a very small piece where the red is illustrated in red here. That's the variable region, and the balance is a constant region. The variable region is specific to each of the individual antigens that this antibody is going to be called on to respond to. The constant region is just like it sounds. It's a series of polypeptide sequences that are found on the same, on the antibodies of each of these classes. So every IgM has the same constant regions. IgG have the same constant regions, but their variable regions are going to vary from antigen to antigen. And there can be thousands of combinations, thousands and thousands of combinations of amino acid sequences that are going to create these variable regions. When antibodies encounter an antigen that they were specifically created to target, the antigen and antibody binding is said to be specific. It's a specific response. The antibody is produced specifically to that antigen. It's not to say that all antigens are one of a kind. Alloantigens, like our A and B antigens of blood type, exist across all humans. So not everyone is, of course, a B type, has a B antigen on their red blood cells, but all humans that have B antigens that make them a B blood type have the same antigen. And those are going to react across with antigens from another human with the same antibody to that antigen. We've talked also about heterophile antigens, where antigens from another species are really so structurally similar that they're going to cross-react in producing an antibody that will react to both antigens. So let's say the cross-reacting antigens of a heterophile antibody is a carbohydrate that's really simple, and it's found on lots of different structures, or it may be a protein with a very similar structure. For example, we can see some, let's see if I can do this. I can't. Let's see if I can write. I can't. I was going to write, it's easier than, that's funny. Let's pretend I can write, but I can't. Heterophile antigens, a great example of kind of a cross-reacting allergen would be something like there's an allergen in latex and bananas that elicit the same immune response. They'll both respond to the same antibody. So that's a heterophile antigen that two different antigens from two different species can both react with the same antibody. Looking at this, we think of the antigen-antibody binding process. We've talked about it in the sense of a lock and key fit. After antibody is produced and it encounters the antigen or epitope, the binding, we call it a lock and key. A really good fitting key is one that's a very specific antigen. Kind of a weak or a poor fitting lock and key could be something like a cross-reacting antibody. It may not be a perfect fit, but it may kind of jiggle the lock. Um, it's a poor fit. Sometimes those cross-reacting antibodies or a poor fit antibody results in an antibody that just isn't going to stay fit, like the key in the lock. And those we're going to see as we go along, how those poor fit antibodies are not really that effective. They can be. We can overcome them by the production of certain types of antibodies that just sort of by brute force will have an effective immune response. But really the best fitting antibodies, the ones specific to the antigen that it was created for, is the best for immunity. I'm just going to say that we're going to remind ourselves that, and we're going to talk about this as we go through the semester, so I just want you to put this away in your pocket, that in our serological tests in the laboratory, cross-reactivity between antigens and antibody is a severe limitation to many of the tests that we see, and that they are not necessarily going to, it's going to drive the way we do some testing in the lab. So as I said, you can visualize the antigen-antibody reaction as that of a lock and a key. A key that just doesn't fit in the lock is said to have, no fit. That represents an antibody that was not created for the antigen. So if this is the antibody and here's the antigen, this antigen doesn't fit into the antigen binding site of the antibody, because this antibody wasn't created for this antigen. It's a no fit. And so if a circulating antibody encountered this antigen, it would pass it right by. It may try and bind to it, but it would know that it's not a binding site. It would know that it's not an antigen that it's looking for, and off it would go. It would not bind off to this antibody. So this is no fit. That means there's going to be no response between this antigen and this antibody. A poor fit of an antibody-antigen reaction is, like I said, what we see in cross-reactivity. The antibody and the antigen may bind, but you can see it's not a perfect fit. This is very, ovally shaped, and this is a little scraggly here. In the end, this antibody may bind to that antigen, but it's really not going to get stuck. The antigen may not bind to the antibody really smoothly, and that binding can come undone. It's not a really effective immune response. We've seen cases where, though, it's better to have, a poor fitting antibody, a poorly specific antibody, than nothing. And I think that we can all remember back to, the first lecture that we had where we talked about the, Jenner's cowpox, smallpox example. Where we found that Jenner had found that the milkmaids had a lesser response to smallpox because they had developed antibodies to cowpox that were very similar. For example, if this is smallpox, this is the antibody to cowpox, they do bind together. So there was some measure of protection for people. It wasn't a perfect fit. It didn't eradicate smallpox in the individual. It didn't mean they didn't get maybe a milder form of it. But to some degree, the cross-acting immunity of antibody production was effective. So maybe they got a lesser version of the disease, but didn't die from it. A good fit antibody represents an antibody that's created especially for this antigen. So this one you can see it's a good fit here, and it has the little spikes here that fit into the V. So this was an antibody that was created exactly for that, and that's a good antibody to fight this foreign pathogen. So when we're talking about antibodies, we're talking about when B lymphocytes get stimulated by antigen. B cells undergo differentiation, and the end product is immunoglobulins. Antibodies are formed or are found in the liquid portion of the blood inside of humans. It's called plasma. Though in the laboratory, we generally use serum for our tests. And what's the difference between serum and plasma? Just to be clear, serum, when collected, is collected in a type of tube where the blood clots within it. And serum is the same as plasma with everything except for the clotting factors that are required to make the red blood cells clot together. I think we'll try and remember to do that in another lecture and have some illustrations of this. Hopefully you've encountered this a couple of times in lab classes already, but if you haven't, we'll make sure that we do that in a future lecture. So chemically speaking, though, antibodies are, mostly polypeptides, which are really, those amino acid chains that we've spoken about before, the amino acid chains that make up all or part of the protein molecule of an antibody. In the cases of antibody, it's mostly polypeptides with a little bit of carbohydrate, maybe about 20% of the antibody. I apologize, maybe 2 to 14% of the antibody is made up of carbohydrates. And in general, antibodies themselves, the production of antibodies in the host, represents about a fifth of all the proteins that we have in our body, in our plasma, circulating plasma, not in our body, good gravy, circulating in the plasma. So 4-1 beta lymphocytes are stimulated to differentiate and produce. They're found in the plasma portion of the host. They can also be bound to the membranes of B cells. We saw that in the last lecture, how immunoglobulins can bind off to B cells as they circulate. Mostly polypeptides, a little bit of carbohydrates, and again, those 5 classes that we're going to start to become very familiar with. Not to put too fine of a point on it at this point, but, just remember that antibodies are essential in the humoral response by a couple of mechanisms. Antigen recognition, and in kind of the biological activities of the immune response that can be enhanced by the action of antibodies. So they can include something like opsonization, which is coating a pathogen with a protein that makes it more susceptible to phagocytosis, and complement activation, which we're going to talk about this in an entire chapter. I think it's in the next 2 or 3 chapters, where activation of this process within the host results in ultimate destruction of the pathogen. So this antigen recognition is how we bind off is how we result in the production of antibody and killing of the pathogen. We're going to quickly veer off from our straightforward talk about antibodies for just a minute, and talking about, really, we just mentioned that about 20% of the plasma proteins are antibodies. But if we look at all the proteins that we find in the plasma, in the laboratory, we can separate the proteins of the plasma using or serum by using a special process we call electrophoresis. We're going to look at a video of this in just a second, but how this works is serum gets placed in, I wish I could draw. It's been established I can't draw with a pointer, so we are going to look at a video in just a second. But so if you have let's say this is the gel that we put in, there's going to be some wells over this side. Oh, holy cow. And we're going to inoculate the gels of let's go back. A small sample is going to get inoculated into some wells over here that I cut into a polyacrylic gel. And we're going to apply a negative charge and a positive charge to the gel. There'll be a negative charge on this side and a positive charge on this way, and it's going to be attached to some kind of an electrical source like a battery. The electrical charge of, proteins and all molecules have an electrical charge of some sort. And the electrical charge of the proteins move them away from the negative charge of the battery that gets attached here. So they start to migrate through the gel, and they start to separate based on their electrical charge. And as they start to move into the gel, we're also going to see how they migrate differently through the gel based on the size of the particle or the size of the protein. Larger proteins move very, very slowly, and smaller proteins move more quickly. The region that we're most concerned about in immunology is this gamma region, this right here. This is the gamma region of proteins. The gamma region, the gamma proteins are basically pretty neutral, and immunoglobulins are in here. Ig is stands for immunoglobulins. So immunoglobulins fall into this gamma region, and they're basically very neutral proteins, so they don't move far away from the electrical current, the negative electrical current. And similarly, they're very big proteins, which are very complex, so they're kind of slow and stodgy getting through the gel, so they don't move very far. So the region that is closest to the point of inoculation or point of origin are going to be the gamma region. There are going to be four other protein patterns that we see here. This protein right here is called albumin. This is a very high concentration in the plasma. It's the fastest moving, and it's the furthest moving. We also have alpha-1 proteins, alpha-2 proteins, and beta proteins. We're not going to be concerned too much about these right now. We will be a little bit more so when we get a little bit closer to some of the laboratory testing that we're looking at. What we're looking at, again, really are these, kind of large, cumbersome neutral proteins that are not moving very far in this electrophoretic pattern. Just a reminder, in case during the video we see it, the negative charge, so remember, this gel is going to sit with a solution in it that's going to help it's going to help the electrical charge to be applied to the gel here, and we are going to turn on the battery, and we're going to see the pattern move across. The cathode is the negative charge, and the anode is the positive charge when we talk about it. So I do need this hyperlink, I don't think is working. If you want to, let's see, I don't think it does. I tried very hard to get it to, and it doesn't. So let me just go out. I have it all queued up. If you want to look at it again at home, you can cut and paste this. There are a ton of YouTube videos on this that we will see. Let's discard that, sorry. That we'll be able to see. And you are going to do some electrophoresis in clinical chemistry when the time comes. We're going to do it during DNA extraction in molecular testing. So this is a quick video just looking really at the super basic process of electrophoresis. And so that really is a super simple explanation of how the proteins are moving in electrophoresis. And you really want to be familiar with this electrophoretic pattern of protein migration, and understand which, the region that we're concerned with oh, hello. There we go. Let's close that off. Hello. There we go. Sorry about that. Kept talking. We're just concerned really about this gamma region when we're talking about this right now. All right. So as we continue, yeah, I think we're all set there. So the illustration here shows the basic structure of antibodies. Again, that blue chain is the heavy chain, and the red is the light chain. The pale blue and the pale pink represent the variable regions of both the heavy and light chain. And this right here at the top is the antigen binding site. So this is going to pretend this is the epitope, the small piece of there's the entire antigen, and on its surface it has little protein molecules that are specific in their structure, and the antibody is going to create a very specific binding site to this. In this case, it's a very simple illustration of the binding site. The heavy chains are always identical. These are, they're just always identical. And so in this case, remember, we're going to have different types of heavy chains that are going to create the properties of the IgG, IgM, IgE, etc. But we're going to remember these two heavy chains are always identical, and these two light chains are always identical. These chains are held together by what are called disulfide bonds. You can see the light chain is bound to the heavy chain here. The light chain is bound to the heavy chain here. The heavy chain is bound to each other through a couple of disulfide bonds here. Generally, we're going to see some variants of this as we go along, but there are disulfide bonds here. And this entire portion right here, we're going to call the hinge region. We're going to talk about that in just a few minutes. So I just said that the heavy chains are identical. Those have the same amino acid makeup. The constant regions of these two heavy chains are the same. The variable region of these two heavy chains are the same, but specific for this antigen that we're gonna interact with. And these, as I said, are going to be either, these are going to be one of several different kinds of chains. We saw in the last lecture how the pre-B cell produces mu chains in its cytoplasm, eventually bound to the membrane, and those are going to make up the IgM molecule. There are similar chains that are going to be developed for each of the other kinds of antibodies. We'll talk about that in just a few minutes. But I want to make note that the light chains are also identical. However, there are a couple of options of them. Light chains can be either kappa light chains, so they're going to be made of kappa chains, or lambda chains. The light chains have to be identical, but for example, we could create an IgM with 2 heavy mu chains and 2 lambda chains, or we could create an IgM molecule with the same 2 heavy mu chains here, but 2 kappa chains. Did I just say that? So this could be kappa and kappa, or this could be lambda and lambda, and still both be an IgM molecule. It's important to note that you never have a mixer, you never have non-identical light chains. So you would never have on the same molecule a lambda chain and a kappa chain. That would never happen. They're always going to be identical forms of chains. But which one it is as long as they're identical, and it's the heavy chain that's going to ultimately drive what kind of a molecule is created, what kind of an antibody. Oops. So we said that there were the heavy chains. We are going to have the production of gamma heavy chains that are going to create IgG. We're going to have delta heavy chains that are going to create IgD. We're going to have alpha chains that create IgA. We're going to have mu chains that create IgM. And we're going to have epsilon chains that create IgG. Those are the heavy chain types that are going to develop in order to create an immunoglobulin. So it's going to be two of these heavy chains and two of either 2 kappa and 2 lambda. So the antibodies basically have the same structural detail in this picture. It's got just a little bit more information in it, including identification of the hinge region, which allows for conformational changes to take place. So let's look at what we see here. Again, we have a unique variable region here, which is going to be the antigen binding site. This part of the light and the heavy chains are unique. They're the variable regions, and those are what provide antigen specificity of the antibody molecule. There are one or more constant regions. The constant region of the light chains are here. Constant region of the heavy chain are here. Oops, I'm sorry. The light chain and the heavy chain here are constant regions. And as I said, the constant regions are exactly that. They're going to have the same amino acid structure. There can and will be different regions within the constant chain that we're going to talk about in just a second, but they're constant in nature. We also see in here the place called the hinge region. So the hinge contains a process in order to be able to have the antibody change its configuration in order to do some binding. So I'm going to give you an example of why this hinge is important. And without me being able to do contortions in front of the class, I'm going to try and describe it to you in terms of a game I hope you all know. Hopefully you know the game of Twister, where you have the board on the floor, the plastic mat on the floor, and it has 1, 2, 3, 4 or 5 rows of colored dots lined up in columns and rows. So there's probably a total of 16 or 20 dots, large dots on a mat on the floor. Imagine you're the antibody. This is your body, and here are your arms. You're putting your 2 arms out in the air, straight up in a Y shape in front of you. So there's your little head right there. So you've created a Y shaped like an antibody. And your right arm needs to bind to an antigen in the upper right-hand corner of the twister board. So you bend down, you put your right hand on that upper dot of the twister board. But this antigen binding site finds an antibody way behind you and to the side. And this antibody has to twist and bend to reach the antigen that's located way down here. If you look at this conformationally, you would say to yourself, how would this work? Well, this hinge right here can allow this arm to twist and conform so that the antigen binding site could hit an antigen epitope way down here. So this piece is able to flap over, and you are able to change the conformational appearance of this antibody. That's the point of the hinge region. It's a really important piece of the antibody structure. Again, the disulfide bridges are going to bind the light chains and the heavy chains, and we know that the antigen binding site is responsible for the binding. And the heavy chain the constant chain regions of both the heavy and light chains are really, those are going to be the determinant structure that tells us whether it's going to be an IgG, an IgE, an IgM. So the constant region talks to us about the ultimate type of chains that are produced in the process. This illustrates the antigen binding capacity of the variable region of the antibody. Again, we're seeing the same variable regions of the light chains, variable region of the heavy chain, and identifying this as the antigen binding site. So if you imagine this is a lock and key, you have got this super specific antigen binding property like this. If I had that same round antigen binding, I had the same epitope on that round binding site that we've seen in this picture. If our epitope looked like this, that antigen would never bind into this particular antigen binding site. It would be a no fit, right? So we have to see an antigen with this conformational shape that's created for which an antibody has been created exactly to fit this. It's a very, very specific antibody, and this would be a perfect fit antibody for this antigen. And so this illustrates just how these antibodies have to be created for very specific structures on the antigen surface. And again, this is not the whole antigen generally. There's going to be, a process here. This part right here, let's say this is the whole antigen. This part right here, this shape that binds here, that's the epitope. That's the antigen determinant. That's what makes this whole antigen look foreign to this antibody. So this green part right here is the epitope. And if we look at the antibody, this is the antigen binding site. Does that make sense? If it doesn't, make sure you come see me, shoot me an email. It's important that you understand the concept of how epitopes, small regions of an antigen can develop a perfect fit. If we looked at this as an entire antigen here, this could be an epitope of this antigen, right? It's a different shape, it's a different conformation, and if this is antigenic in nature, we could produce an antibody to that as well. And that would look more like the last picture that we had. It would almost look like that would be the antigen binding site for, this particular antibody. So that brings us to specific antibodies. So we've seen now kind of the general processes by which we create antibodies through B and T cell activation. We have seen now the structure, the general structure of antibodies. We understand the lock and key fit of antibody-antigen reactions. And we understand the conformational changes that can take place in antibodies in order to in order to be able to fit with antigen determinant sites of something foreign. What do we need to know now? Let's look into each of the different antibody classes in a little more detail. We're going to spend just another couple of minutes looking at the components of the antibody in a little more detail. First up, we're going to look at the heavy chains. As I said, there's 5 heavy chain possibilities. We saw in the B cell during the B cell differentiation that the mu chains are synthesized in the pre-B cell, but other chains can be programmed for production. They're programmed genetically for production. We just happen to see the example of a mu chain during the production. But again, genetics and processing will determine what kinds of heavy chains need to be produced. As the heavy chains are synthesized with variable and constant regions, It's the constant region, as I said, that is going to determine which heavy chain will be unique to the class of immunoglobulin molecules. So the heavy chain, that constant region of the heavy chain is what makes it either a gamma chain, a mu chain, an alpha chain, a delta chain, or an epsilon chain. That's the constant region is responsible for what kind of chain that heavy chain is going to be. Each of these chains types, the gamma chain, mu chain, alpha chain, delta chain, and epsilon chain, represents what we call an isotype, or a unique amino acid sequence that's common to each class of immunoglobulin. So there's the amino acid chain of the amino acid sequence of a heavy chain is called the isotype, and that's going to determine that we're producing a gamma chain or a delta chain, whatever. So that's an isotype. And if we look at the next slide, we can see that the structure again is one where the complete heavy chain is very large. It has anywhere between 450 and 550 amino acids in sequence. And it's going to be that sequencing that the differences in sequencing is what makes different isotypes, different types of heavy chains. The delta chain, the gamma chain, the epsilon chain. As I've said before, both heavy chains are identical, and heavy chains contain both the variable region and the constant region. The variable region is a small portion, about 110 of those, roughly 500 amino acids are in the variable region, and the rest is the constant region of the antibody. As I said, the isotype is the amino acid sequence of the H chain that gives each immunoglobulin class its own specificity. As the heavy chains are produced with variable and constant regions, we can have minor variations in that amino acid sequence of the constant region that gives small differences to a particular type of immunoglobulin. For example, we can have, pardon me, we can have an isotype with, we have 3 examples of different variations in chains. So we can see this is the isotype where this is for, let's start back. This is the isotype version. So if we look at this, we have the same amino acid sequence on everything. We have, we've created either a gamma chain, a delta chain, a mu chain, whatever. We can have little minor variations in the constant region. We might change up the amino acid sequence here, but they're going to be changed equally. So both chains are going to be mirror images of each other. And when we have little variations in amino acids within this constant region of the chain, that's what we call an allotype. There can be the purpose of these variations is going to help to create these heavy chains to have, a unique, it's going to ultimately lead to a unique binding site. And so we see here there can be little changes in the constant region down here or the constant region of either the light and the heavy chain. And these are going to help define ultimately what that antigen specificity is going to be. We're going to see in a minute that there are 4 different allotypes, 4 different minor variations that are going to cause some conformational changes in IgG molecules, IgG subclasses. IgA has 1 subclass, 1 variant, and kappa and lambda chains can also have some variants. And let's see, the idiotype is variations in the variable regions, and these changes in the amino acid sequencing in the variable regions, that's what really gives us that super specific antibody response to a specific antigen and creating that unique antigen binding site of every antibody that we produce. So by definition, I just want to make sure that you understand the difference between an isotype, an allotype, and an idiotype. Isotype is the sequencing of the constant region of an H chain, determines what kind of an immunoglobulin class it is. Is it an IgG, an IgA, an IgM? The allotype are just minor variations in the constant region of either the light chain or the heavy chain. And this is going to represent some subclasses of each type of immunoglobulin. And we're going to really look at the subclasses of IgA and IgM as having subclasses. I'm sorry, the class of immunoglobulins of IgA and IgG have subclasses. IgG is going to have 4 subclasses. IgA is going to have 2 subclasses. There can be subclasses in the kappa and lambda, the kappa light chains as well. Although we don't see that in our processing that frequently. And the idiotype really is just referring to all of the, you can have thousands of different types of IgG molecules, but the idiotype of it is saying each of those thousands of IgG antibodies are unique for its own antigen. It has its own specific antigen binding site. We've talked a lot about heavy chains today, so let's take just a couple seconds to look about the specifics of light chains. Light chains, took a long time for them to figure this out. A long time. It was in the 1800s ultimately that they figured out to some extent that light chains existed, but they're very small amino acid sequences. 200 amino acids for the entire light chain. When we remember the heavy chain is probably double or double and a half, up to 550 amino acids in length. So the light chain has also a variable and a constant region, just like the heavy chain. The variable region and the constant region of the light chain. But they were very, very difficult to find out this, and it was really important to do this so we could start to look at the structure of antibody production. There was a doctor, Dr. Henry Bence-Jones, and that name is going to be important because we actually call light chains Bence-Jones proteins. They were named after him. And when he was studying proteins that were secreted in patients with multiple myeloma, he realized that he was able to identify them through a process where he found that there were proteins being secreted into the urine by these patients. He heated the urine to about 60 degrees Celsius, and the Bence-Jones proteins precipitated out. They formed a precipitant, or they aggregated together and kind of fell out of solution as a big particle. And he further heated it, and they redissolved. So he was able to take a large quantity of urine of a patient that was secreting these specific antibodies. He was able to, or these proteins, they were secreting these proteins. He was able to heat the urine, have the stuff fall out of solution, pour off the other stuff, reheat it some more, and it went back into solution, and then he could test it. So he was able to identify these light chains, and was able to look at them and be able to help us determine kind of their structure. As we've discussed, there are the 2 variants of light chains based on substitutions in the amino acid sequence, and we can determine that the 2 different kinds of light chains we can have are either kappa or lambda. And just as a reminder, always 2 kappa or always 2 lambda. We're never going to mix them up. Kappa and lambda chains are found in all 5 classes of immunoglobulins, but only one is present on any given molecule. So Again, this could be kappa chains or lambda chains in creating this immunoglobulin. And there can be some, let's pretend this is an IgG. Some of them can be kappa, some can be lambda, and it's going to be based on this constant region, the amino acid sequence. So these light chains are small. If we look at them comparatively to the size of the heavy chains. So it's really what I want you to know about the light chains. Light chains are just as important in the process as heavy chains in that they aid in developing that antigen binding site that's going to be so important for each antibody produced. We look at antibodies on a page in as a very two-dimensional structure, but it's really not. It's basically 4 chains, 2 heavy and 2 light that we've seen in our little 2D conformation. So we have 2 heavy and 2 light. So it's a 4-chain structure. But instead of existing like little Y's circulating in your blood, it's really folded into the chains are wrapped around and folded up into 2 kind of globe structures. Here, this represents what the variable domain looks like. So this is equivalent to this area. So there's that. And then the rest of it, this constant region here, oops, if I can get my pointer, this constant region is really reflective more in a conformational structure that looks like this. So those 4 chains are going to be bound together, and they get folded into these really kind of compact structures that this looks like a little globe at one end of the structure, and this looks, like a globe at the other end. The antigen gets actually captured within this globe structure, and they call it a barrel, and it binds to a number of amino acids within this structure that are called hypervariable regions. I don't see that I put it on there. Hypervariable regions. That's what's going to make this very specific for an antigen. Those hypervariable regions are the antigen specificity of this structure. The constant region, instead of really being just like a, kind of a straight line, actually ends up being folded back and forth, back and forth. You can see it's represented here, how it's folded, folds up, folds down, folds up, folds down, folds up, folds down. And they call that the beta pleated sheet right here. This is what gets held together in the constant region by those disulfide bonds. So the peptide chains of the constant region get folded into it's called beta pleated sheet, and that's the constant region of both the light and the heavy chains get folded like this into one large structure. What else do we need to know about this? So this is the barrel. The antigen gets bound into it, kind of cupped into it, into a very specific binding sites. There are 3D read. You can see on here this CDR region, and there are 3 CDR regions per heavy chain and per light chain. So there are a total of 6 because we have 2 light. We have a set of 2 light chains and a set of 2 heavy chains. So we have a total of 6. So there are 6 of these CDR binding sites. And it really takes just little teeny changes in the amino acid structure of the variable and the light, the variable region of the heavy and the light chains to create these different CDRs. They change the configuration of this entire binding site. And these CDRs are going to be ultimately these 1, 2, 3, and then we'd have, if this represented the light chains, we'd have 1, 2, 3, 4, 5, 6 for heavy and light chains. And those are really going to be the binding sites for the antigen. And each one of these vary from this antibody to another antibody to another antibody by little teeny changes in the amino acid sequence of each one of these things. That's how we're going to get specificity. It's just changing sequencing of amino acids that are going to generate the specific antigen binding site for this. Because we have a fair number of these CDR regions where you can have minor changes, the number of antigen binding sites that can be possible for synthesis is very large. And that's how we create antigenic diversity, is by creating many antibodies with very, very small conformational changes and structural changes that changes that antigenicity, that antigen binding site. So we talked about in one of the first two chapters about how important it was to have the ability to recognize many different kinds of antigens, and that when we have bred out the capacity to develop multiple antibody production, when we have, in the case of the Florida panther, the gene population was so tightly bound that the genetic capacity of the what did we decide it was, a pride maybe, that the small group of panthers had so little genetic diversity anymore that they weren't able to respond to new pathogens in a way that was effective. When they brought in new females into the pride of panthers, whatever it is, the group of panthers, they brought new genetic diversity in, and they were able to develop the genetic capacity to recognize more antigens again, for lack of a better word, or the right word, the pride of panthers was allowed to flourish. So having a strong genetic capability to be able to produce different types of antibodies and to produce a robust antibody system is really important to the long-term survival of a group of individuals. And finally, before we look at the individual classes of immunoglobulins, let's take just a minute to look into the structure of the antibody molecule and really what the activity of each region means in terms of its function. Antibodies can be treated with proteolytic enzymes that cleave or cut the immunoglobulin into 3 different fragments. And this helps identify the functionality of the regions of the Y-shaped immunoglobulin. 2 enzymes are used for this purpose. One is called papain and one is called pepsin. And 3 different fragments can result from cleavage. The first is called the FC fragment, or the fragment crystalline. The next is the FAB fragment. This is the fragment antigen binding. Well, you can imagine where that is. That's in the variable region, right? And we can have the FAB2 fragment, or the fragment antigen binding 2. And we're going to see these in just a second. And again, the purpose of this process was to determine what each of the individual portions of the antibody do and how we can use them in the laboratory. So if we look at papain as the enzyme that is going to cleave the antibody, so here's our antibody, pretend it was still all bound together. Papain comes in and breaks the disulfide bonds at the hinge region. Remember, we had the little hinges here that would allow these arms to move. Papain breaks those, and because we broke them, the hinge region here, we end up with 1, 2, 3 different pieces. The immunoglobulin breaks into almost, basically 3 even pieces. This is about, between 45 and 50,000 Daltons, 45 and 50,000, 45 and 50,000. So we've broken into two FAB regions, 1 and 2, and one FC fragment. This is the fragment crystalline, and this is a result of papain. The FAB regions each contain 1/2 of the heavy chain, right? So here the dark blue is our heavy chain. So each of the FAB contains 1/2 of a heavy chain and 1 full light chain. These two identical, oh, and they're still held together by disulfide bonds. So we maintain the integrity of this antigen binding site, which is really helpful for us because the FAB portion is maintained in conformation and the binding site remains intact. These two identical FAB fragments are found to have antigen binding capacity. So that's how we ended up with the name of FAB, the fragment antigen binding. The variable region is, you remember, that's the lock and key piece of the model. So the fact that we can maintain this intact and still bind is going to be really useful to us in the laboratory testing that we do. So the FAB fragments, as I said, 1/2, 1/2, held together by a disulfide bond, contain the variable region, and that's what we see. Let's see. The second piece that was in papain digestion, so we had 1, 2 FAB units, and then we had 1 FC unit, the fragment crystalline or crystallizable. The third fragment is really the constant regions. It's the major portion of the constant region. Of course, there's still constant region up here, but it's a smaller amount of constant region. So this really reflects the constant region of the immunoglobulin molecule. It contains 2, it has no antigen binding capacity because clearly the antigen binding is up here, not in the FC region. It has no antigen binding capacity. It really is looking at what's called the carboxy terminal half of the H chain, and it's still held together by disulfide bonds, right? So it's still, this piece remains intact, and it was it contains the portion of the the portion of the immunoglobulin is the heavy chain, and that's going to be the part of the heavy chain as we remember, that's going to determine the isotype or the class of immunoglobulin, whether it's going to be whether these heavy chains are delta or epsilon, and it would make this entire molecule, you know, IgG or IgE, for example. So this is the constant region that specifically makes the class of antibody that's going to be produced. So that's an important understanding of this. The purpose of this portion of the molecule has really two processes that are involved. So the FC region is important in what are called the effector functions, the actionable functions of the immunoglobulin. The first major one is that this area of the immunoglobulin is responsible for what we call opsonization. And we've talked about this before, that opsonization is really just a coating of the foreign substance with some proteins, the serum proteins that are going to attach to the foreign pathogen and make it more susceptible to phagocytosis. We're going to talk a lot about that. The second function of the FC portion of the immunoglobulin is what is called complement fixation. We're not going to spend a lot of time on it now, but just so you know and start to get a sense of the term, complement fixation is it's a test methodology that we use where we bind a naturally existing serum protein called complement, and we're going to bind that to an antigen antibody complex and see the process that happens after that. The ultimate role of complement in the host is to kill the pathogen. So we're going to be able to use complement fixation within our test methodologies to be able to look and see if an immunoglobulin will, quote unquote, fix complement. So, but don't get too hung up on that now. Just really remember that it's a process in the laboratory testing where we bind that protein called complement to an antigen antibody bound complex. So the antigen antibody bind at the binding site, and then we're gonna attach complement to it and see what happens. That's the point of complement fixation. So I said there were two enzymes that we could look at. We could look at papain. We just saw how that cleaved the molecules or enzymatically broke an immunoglobulin down. Let's look and see how pepsin digests and cleaves immunoglobulins. And they do cleave them differently. Papain, and like its counterpart, or the other enzyme that we reviewed, pepsin will also result in cleaving into essentially an FAB region. You can see though that instead of cleaving here at the hinge, pepsin will cleave so that it cleaves at the in such a way that the disulfide bonds hold the two FAB portions together into one large unit. So we call this an FAB2 unit. So we have two bound FAB portions. So that's called the FAB2 portion. So the two antigen binding sites are still held together by that disulfide bond. Pepsin also has the capacity to break the FC portion instead of seeing two large heavy chains that are still bound together by disulfide bonds, and actually is going to break those disulfide bonds and end up with little teeny pieces of digested material here from the heavy chain. So like its counterpart from papain digestion, this FAB2 region is really useful in the laboratory in that it can still bind with antigen in the tests, but it has one extra capacity, not only to bind with the antigen, but because it's a large molecule, we can actually see that complex when we bind antigen here. It can fall out of solution and we can test it. It's a way to be able to visualize that antibody-antigen reaction. So the fact that this is a very large molecule makes it really helpful in the laboratory. Let's see, when I say it falls out, it means that, when the antigen binds into the antigen determinant site or the antigen specific site, creates a heavy molecule, and it falls to, it falls out of solution into sort of clumps and makes it a visual capacity. We may need to add it to a carrier molecule or something, but it's very, very helpful. The FC region in this case in the laboratory is not super helpful to us because it's really bound into very, very small portions of the heavy chain. The heavy chain is broken into FC, and so you'll see it just has the little apostrophe sign after it. That's just telling us really these are structurally different than when we see in papain. So that's really all I want to say about the enzymatic piece, and I just wanted to do that so that we could start to work into our last phase, which is looking at each of the actual antibodies that are produced. So yay, after all of this background, all of this review, the background of the structure of immunoglobulins, the function of immunoglobulins, the pieces of immunoglobulins, the variable, the constant, the idiotypes, the allotypes, all of that information behind us, we finally get to, what's like the prize of the chapter, which is antibody production. I'm going to take a 2-second break here, and I'm going to put us on hold for just a second, and then we'll start right up. And so we are going to look at the 5 different classes of antibodies. Let's look at them each. Let's look at them structurally first, and then we'll look at them individually. Oops, here we go. There we go. All right. Antibodies like IgG, IgE, and IgD are what we call monomers, or one structural unit of the Y shape immunoglobulin. Each of these has 2 antigen binding sites. So here's an IgG molecule and has 1, 2 antigen binding sites. Similarly, IgE and IgD. That's the same structural. You can see, though, there are differences in, for example, the hinge region and how many disulfide bonds there are. So there are structural differences between them, but they're, this has a different number of constant regions. So there are definitely differences in their structure that make them unique antibodies. But they have the same basic conformational shape with the same number of binding sites. So IgG, IgE, and IgD have 2 antigen binding sites. IgM, on the other hand, is a really large molecule that is a combination of 5 monomers, all bound with chains in between them that we're going to see in a few minutes, that bind them together as one large pentamer. This pentamer has 10, because there's 5 individual subunits of IgM. It has 2, 4, 6, 8, 10 binding units. So this has it's a 10 binding unit molecule. Very big and very that large size and the large number of binding sites is going to be very useful to us in a few minutes when we talk about it. And IgA can exist in 2 forms. We can see it as a monomer if we imagine, it's cut off here. We can see it as a monomer, but it's not pictured here because really the unique version of this is this dimer. This, 2 units, this 2 structural units bound together, what's called a J chain or a joining chain. The IgA also has this kind of squirrely S-shaped protein running through it that we're going to see in a few minutes. It's called a secretory protein, and that's going to be also very important in its structure and how this is formed as a unit. So again, IgA can be in a monomer or a dimer form. We like to think of the dimer as, kind of the interesting one of the immune system. We'll take a look at it in a few minutes. But we're going to start with the granddaddy of all the immunoglobulins, the IgG. Why is this such an important immunoglobulin for us? It's the most abundant in all the serum immunoglobulins. It's not necessarily, it isn't the first immunoglobulin that's produced after a challenge by an antigen and production from B cells, but it is, it has the longest half-life, and so it sticks around for a very long time. What are some of the things we know about IgG? It's found both intravascularly and extravascularly. So it does travel through the bloodstream and out into the tissues. It's a single structural unit that we just saw. And we know, because we've talked about it today, that it has the gamma chains are coded, the heavy chains are coded as gamma chains from the constant region. Functionally, what do we know about IgG? Because it's a monomer and the size of IgG can actually cross the placenta from the mother to the infant, and so this is the major immunoglobulin that provides immunity for a newborn. IgG is able to remember we just talked about that protein that circulates called complement, and that the ultimate end product of the complement system within human beings will result in lysis of pathogens. Well, IgG is capable of binding complement, and it's going to activate that complement system. So we're going to talk about that in another lecture in great detail, but for now let's just remember that IgG crosses the placenta. It can bind complement, and it also has a capacity to provide opsonization. So remember, opsonization is just the immune process where particles like bacteria will be coated by a protein and be targeted for destruction by a cell like a phagocyte. So opsonization is a method of targeting an invading particle to be killed, right? So IgG has a capacity to coat the pathogen and signal opsonization, signal for phagocytosis. IgG also has a unique capacity that it's able to help neutralize toxins and viruses in a way that other immunoglobulins cannot. In the laboratory, IgG as a test methodology is a really useful tool for tests that we are creating in the laboratory. Most of the serological tests that we're going to talk about, a vast majority of them, let's say maybe not most, a vast majority, are involved in what we're going to see are called agglutination or precipitation reactions. Agglutination really just means kind of clumping together, making a clump of larger particles like red cells. And so IgG is a really good and effective mechanism by which we can cause those particles to clump together. And precipitation reactions are one where there are small particles in the liquid part of the blood. Remember, they're soluble. Soluble means they're dissolved in the blood, that get brought together and become visible in the test reaction when they bind to immunoglobulins. While IgG is able to both agglutinate and precipitate, IgG is a better precipitator than agglutinator. So we're going to see some test methodologies. IgM is a great agglutinator, but IgG is, while it can agglutinate, there are going to be some forces that keep it from being as effective as it could be. And we'll talk about those in a few weeks, but just remember that IgG is a great precipitator. It's a great little binder that helps bring together some of the soluble proteins in the lab. Again, we're not going to do these all today because, frankly, I have to have something to talk about in the future. But just the general characteristics of each of the immunoglobulins is important to understand. As I mentioned earlier, there are 4 subclasses of IgG. There's IG1, IG2, IG3, and IG4. And if you look at the illustration here, how do they differ? They differ in the conformation of the hinge region and the disulfide bond. So IgG1 has 2 disulfide bonds. IgG2 has 1, 2, 3. I can't see, 3 or 4. 4, I think. IgG3 has like a large ladder of disulfide bonds, and IgG4 has, a different hinge region. But in reality, the point of each of these conformational changes, each of these structural changes, is that they allow the IgG subclasses to bind to different kinds of antigens. IgG1 and IgG2 respond to IgG1 and IgG3 are great responders to protein-based antigens. IgG2 and IgG4 respond more to polysaccharide kind of antigen bases. So the subclasses can all respond in the secondary, what we're going to term the secondary immune response, and we're going to look at that in just a few minutes. But they're going to be active in different ways and respond to different immunoglobulins. We are going to see that IgG plays a role in both primary and secondary immunity. But it's not the first antibody that gets produced after the host is challenged by a pathogen. It's a really effective immunoglobulin because it has time to develop really good binding sites on the second challenge by a pathogen. So we'll see that in a few minutes. IgA is really the second most sort of complex immunoglobulin, in that it comes in the form of a dimer and it develops in a really unique way. IgA, of course, has alpha heavy chains, and we're going to see this in the secretion. So we're going to find it in places like the mucosal associated lymphoid tissue. It comes, as I said before, in both monomeric or monomer or dimeric or dimer forms. So we're going to see that the dimer form is the secretory version of IgA. It's the one that's found in secretions, and it's in a dimer form, and it's held together by this joining chain, the little joining chain. So we call it a J chain. It also has this, you see this little squiggly protein here? That's going to be called the secretory component, and we're going to see that in just a second. But before I get off of this stage, IgA is really important in respiratory, urinary, and bowel infection control and response. And it's a pretty large, it's got a pretty decent composition within the total immunoglobulin pool. About 10 to 15% of all the immunoglobulins that are circulating are IgA in form. So let's take a look at the IgA role of IgA. IgA does not cross the placenta, so it doesn't provide immunity to the neonate in that way, but it's present in very large quantities in breast milk, which can transfer into the gut, into the mucosal areas of the infant. So you can see it's very useful in posing protection for an infant in the gut, which is what we're looking for. IgA is found in the mucosal areas. We said that it aided in urinary and bowel infections and respiratory infections as well. So finding them in the breast milk is very helpful. So IgA does not cross the placenta, and it does not bind to complement. So as I said, there are two subclasses of IgA. The first one is mainly found in serum, and that's IgA. It has a basically anti-inflammatory function, and it does what's called downgrading IgG-mediated reactions. So it helps to control some of these processes that IgG has an influence on. So it's going to control excessive phagocytosis, chemotaxis, bacterial activity, and cytokine release. And in that way, that downregulation is what makes it anti-inflammatory. Remember, if you have constant release of cytokines and action of immunoglobulins and other components of the immune system, you end up with hyperinflammatory responses. So IgA subclass A1 is anti-inflammatory. IgA2 is the dimer form of this, and this is the one that we're going to see in present in the secretions at mucosal surfaces. It's a dimer that we find in the respiratory, urogenital, and intestinal mucosa, and its main function is really that of it's almost like that of the innate immune system in that it keeps the antigen from penetrating farther into the body. It will have a very specific response to an antigen, but it does survey the area looking for pathogens. And as I said, it has this little, so not only are the two monomers formed by this J chain, they're held together by that joining chain, but we've got this secretory component called the SC. You'll see it referred to the SC region, or the secretory component. That's going to help make it a little, it's designed to make it a little bit more resistant to some bacterial destruction. And let's see in the next slide how we get to this point. As you can see from the illustration here, IgA, just like all immunoglobulins, is produced in plasma cells, as are the J chains. Those joining chains are also produced in plasma cells. These plasma cells have been hanging around in the MALT, the mucosal-associated lymphoid tissue, and IgA is synthesized and released as a dimer from the plasma cell, because the plasma cell is creating both of the, all the necessary components. So it's actually secreted from the plasma cell, not as a monomer, not as multiple monomers that join up later, but it's secreted initially as a dimer. The IgA dimer gets captured by a receptor on the epithelial cells of the mucosal lining that actually has the SC. There's the SC, there's the secretory component, and it's attached to the receptor on the epithelial cell. And the SC, the secretory component, binds to and attaches itself to the dimer. And the entire thing, that entire complex is by means of endocytosis, bringing it into the cell, processes in, and it moves through the epithelial cell in a process called transcytosis. So it's just moving through the epithelial cell and exits out outside the other end through a process we know as exocytosis, is leaving the cell as a fully formed IgA with a secretory component bound to it. The secretory component is really going to help transport this IgA now out to the surface of the mucosa, and it also helps make this whole antibody more resistant to the actions of bacterial enzymes. So it makes it a very strong antibody. At this point, the secretory form of IgA allows the IgA to patrol the mucosal surfaces and almost it does act like a first line of defense, in that it can do a couple of things that are not direct antigen related. It can neutralize some of the toxins produced by microorganisms, and it can prevent some bacteria from adhering to the mucosal surfaces. So secretory IgA has a really important function in the mucosa. As I said, IgA provides passive immunity to the newborn during breastfeeding, and IgA antigen complexes, when it does complex with an antigen, they get trapped in the mucus and get eliminated by, sneezing or coughing. They get trapped in the mucus, and just as we've seen in the innate immune system, how the cilia gets gets irritated, and it flushes the entire complex out through a sneeze or a cough. Interestingly enough, when you get these immune complexes, these IgA immune complexes, together, and we've got antigen-antibody complexes, they can trigger another form of complement. Remember I told you a few minutes ago that IgG can trigger complement. It can bind to complement and start that process by which pathogens are killed. IgA can trigger an alternate version of complement. It's not the same version that IgG works with, but it will trigger a different version of the complement pathway, resulting in the same endpoint, which is killing of the pathogen. What else can an IgA, secretory IgA do? IgA can bind to some receptors on some different kind of phagocytic cells like neutrophils, monocytes, macrophages, and do what's called a trigger of a respiratory burst. And it results in those phagocytic cells releasing their granules and having the actions of those granules take place. It could be to trigger more phagocytic cells to the area. It could be to kill, to lyse something. So those are some of the examples of what else secretory IgA can do. Both forms of IgA, not just secretory, can act as an opsonin, and we just saw how it can be a promoter of phagocytosis during this respiratory burst. That was IgA. Let's move on to the big one, the big IgM molecule. This is the pentamer. It's really big. It's about 900,000 Daltons, so it's super big, which makes it a little cumbersome, but it's a nice tool of the immune system. So again, we know from our discussion about B cells that IgM consists of the mu heavy chain, and that's in the constant region. We know of the mu of the heavy chain. Oh gosh. It represents about 10% of the immunoglobulin pool of the host. And it has a relatively short half-life, only about 6 days, compared to, IgG being, 20, I think it was around 23 days. Because it's such a big molecule, it's not going to circulate, it's not going to be able to get out into the tissues. So we saw examples of how cells can transfer through, diapedesis into tissues. This is too big. This is way too big. If we think of a typical antigen, being between 10 and 100,000 Daltons, so, and a very small piece of that is antigenic, 900,000 Daltons. So it's really big. So it's restricted to intravascular space. It's not going to be extravascular. It also will fix complement just like IgG does. It does not cross the placenta. It's way too big to do that. And we're going to see in a few minutes how it's really important in what we call the primary immune response. We're going to see that there are two immune responses, a primary and a secondary immune response. The primary immune response are the first antibodies that are produced after activation and identification by B cells of an antigen present. IgM can exist as a monomer. We saw it on B cell surfaces in the last chapter, in chapter 4, but generally we think of it as being a pentamer, and we see it in secretions. Like the IgA, the pentamers are held by joining chains between the two monomers. So here's a monomer, and then there's a joining chain between them. This is a really effective molecule because there are 10 binding sites, 1 antigen binding site, 2 antigen binding sites. 2 antigen binding sites per monomer. There are 5 monomers, so 5 times 2 is 10. That 10 binding site makes this an effective immunoglobulin because it's the first immunoglobulin that's produced after antigen stimulation. And as a result, it's not necessarily a really specific one. We haven't had a great opportunity to develop a really tight lock and key. It's what we call kind of a poor fit lock and key. But there are so many antigen binding sites that it kind of overcomes the lack of specificity of the lock and key, that just kind of by sheer force, it can be effective in the challenge against a new antigen, with this first antibody that's produced. And it provides opportunity for the host to be fighting off the infection while IgG, a more specific antibody, is being produced. Those 10 binding sites are said to have so 10 binding sites means that IgM has a valency of 10. So IgM is said to have a high valency. That just means 10 binding sites. So and as I said, it makes it so even though the lock and key fit isn't great, with a binding force of 10, the weak lock and key is overcome by just the sheer volume of binding sites. We say that, and I don't see it on here, we say that something that has a poor lock and key, I told you it doesn't stay, the antigen antibody doesn't stay bound really tight. That's what we call affinity, AFFINITY. We're going to talk about affinity in another week or two, but the lock and key is what we call affinity, and IgM doesn't have a really high affinity. It has a weaker affinity than say IgG, but that valency, that number of binding sites, will make up for it. Let's see, I just mentioned found intravascularly, can't cross the placenta. It's known as the primary response antigen. Appears first after antigenic stimulation and in the maturing infant. So as infants start to develop their own immune response, it's IgM that they see first. But it doesn't create memory. It really only synthesizes as long as the antigen stays there. So it's not found on memory cells. Memory cells, we want to be having antibody produced that's very specific to the antigen, right? Some of the other functions of IgM. IgM is really known as the most efficient immunoglobulin for this process we called complement fixation. That's because there are 10 binding sites, right? So if the antigen binding complex is what's going to cause the start of that complement process, that complement killing process, then the fact that you've got 10 opportunities for antigen to bind on one molecule of IgM, you can tell that that's going to be a really effective and efficient trigger for complement. So what else do we know about it? It's a fantastic agglutinator. The large number of binding sites, again, makes it really efficient at binding off antigen, right? So it's a great agglutinator. It also has a really long arm reach. If you think of the fact that an IgG is one monomer here, but IgM has 10 of them, kind of the reach of antigen binding site to antigen binding site is wide. It's really long. So it's able to clump up cells or make complexes of antigen antibody in a very effective way. Like we've seen before, it's an opsonizer, great opsonizer, and it also has some mechanisms for neutralization of toxins. IgM has that J chain, so it has some measure of secretory components like IgA, and it too kind of travels along the epithelial cells and can patrol some mucous membranes as well. And finally, we know that IgM, we've seen it develop on mature B lymphocytes in early stages, so it's very early on developing B cells. IgE is an epsilon heavy chain. It has a very small percentage of the total serum proteins, are IgE. It has very, very specific functionality we're going to see in just a minute. It's not produced by plasma cells of the lung and skin. Oh, I'm sorry, it is produced by plasma cells of lung and skin. It doesn't fix complement, and it doesn't cross the placenta. IgE has a functionality in allergic reactions where the Fc portion will bind very, very strongly to cells of the allergic reaction, the mast cells and the basophils and even eosinophils. And what it does, it's a mediator cell, and it causes those mast cells, basophils, and eosinophils to actually release the contents of their granules, including things like histamine and heparin, which results in what we classify as typical allergic reactions. We're going to see this. Don't worry so much about type 1 hypersensitivity. We're going to see a slide on it in just a second. But just be aware that it's present in type 1 hypersensitivity. We're going to spend a lot of time on type 1 hypersensitivity in another chapter. Just getting the last of our building blocks down right now. So we see this increased in allergies through mast cell and basophils specifically, and parasitic infections we see in eosinophil and basophils as well. IgE can trigger neutrophils and eosinophils to play a part in the destruction of big pathogens like parasitic worms that aren't easy to be phagocytized. I showed you the video when we looked at the cells of the immune system where we saw the parasitic worms being the cells going directly to the parasitic worms, where they're going to degranulate and kill the worms directly that way. They're going to have a more cytotoxic effect on pathogens like that can't be ingested fully by a cell. So IgE triggers that response. It helps to draw cells to a pathogen. So it's a really effective immunoglobulin in parasitic infections as well as allergic infections. This is just an example, a picture of what we see happen at the site of an allergic reaction. Things like hay fever, asthma, those kinds of things are type 1 hypersensitivity reaction, and you get things like this, like hives. This is called a wheel and flare hive. That's characteristic of IgE response in allergic reactions. This is the reaction. Type 1 hypersensitivity is the reaction that causes anaphylactic shock that you hear people going into when they're having an allergic reaction. So it's the degranulation of those mast cells and basophils and eosinophils that cause this reaction when you have an allergic reaction. It's quite severe, but it's definitely an IgE function. It's the IgE binding to the allergen, and then binding off to the cell and complexing to the cell that causes this to happen. We'll see that in further detail again in a few chapters. I know I keep saying this, but we've got to build the blocks and have you understand all of the components of the immune system before we can start to see them in action. We're getting very close to the end of you may want to be back building blocks after we start on complement next week. IgE, I joke about it. It's the I don't know. It's the I don't know immunoglobulin. We found now that for the longest time, we really didn't know what it did. Now we've seen some functions that we've seen, and we've talked about them before, that they can bind to the membrane of some of the B lymphocytes, where they can aid in B cell activation and, even extending the life of a B cell, it's theorized. But IgD have the D heavy chain. The heavy chains are produced as delta heavy chains. It's a single structural unit or a monomer. It's a very small amount of the immunoglobulin pool. It is generally cell bound. It's not a free, it's not a circulating immunoglobulin. We usually see it on unstimulated lymphocytes. IgD does not cross the placenta, and it doesn't fix complement. It has a very short half-life, just 1 to 3 days. So that's the end of us looking at the antibodies themselves. Now we looked at kind of their structure, we looked at their function, we looked at the individual immunoglobulin classes. Now how does it work? How do we put this together? We've seen, and we went over at the beginning of the lecture today, about antibody production. There are 2 phases of antibody production. There's a primary and a secondary immune response. The primary response happens on first exposure to the immune response. I think I'm going to go up to the next slide just so you can see. We're going to remember that it has a gradual rise in antibody production, taking anywhere from days to weeks to reach its plateau, and after which the antibody level declines. In the first phase of this primary response, so this is if we think about this as antibody production. So the host has been exposed to a pathogen and has developed an immune response against it. We have this primary response. We start off with a brief lag phase where for 5-7 days you really don't have much happening, after which we start to see immunoglobulin IgM produced. This is, remember, that large, not so specific pentamer that gets produced. And this phase of IgM production lasts about 10 days. And so you can see it starts here at, in the 6, 7, 8 days. And it lasts until, the 16, 17, 18 days, and then it starts to drop off. And eventually at about 45 days, you're back down to baseline. So a few days later, we start to produce IgG, immunoglobulin IgG. Remember, this is going to be more specific than this. It's had a little bit more time to develop, and it has a more specific antigen binding site. It's a better antigen binder. But you can see in the end, the IgG really is produced in just about the same quantity as the IgM during this primary response of the immune system. If the antigenic challenge is done and there's, surveying and we're not finding any more, the circulating IgG and IgM are not finding any more antigen, they drop off and it lays dormant. We're not going to produce any more antibody to that specific antigen. However, if there's a second exposure, doesn't have to be today or tomorrow, it can be whenever. After that second exposure to the same antigen, what happens? The importance of those memory cells kick in. We keep talking about the production of memory cells, especially in the T helper, the cell-mediated immune response, more so than more so than if a B cell is stimulated by an antigen. But the T cells build those B memory cells. And that recognition of the antigen is immediate. So if we imagine this is a subsequent contact with the immunogen, look at the lag time here. It's right away. Bam. You'll still be producing IgM, just about the same amount. But again, look, there's really no lag time. Remember here, the lag time was, 5, 6, 7 days. Look at the lag time now. There's the start, and it's, a couple of days later, and bam, IgM is produced. But at the same time, what's producing? This really fantastically specific IgG. Not only does it produce very quickly now, remember we had an additional lag time in the primary, but look at, not only is it a really short lag time, but it's a huge mounted response. So when we talk about building immunity in a host and the importance of building memory, whether it's by virtue of this primary exposure having been a vaccine or um, a natural exposure to an antigen, that being able to build memory is really important. So you have this really quick and rapid response. The primary immune response, we think of it as primarily an IgM base because it's the front runner. So IgM is the primary, or is the predominant, immunoglobulin that we talk about when we talk about IgM, because it responds first. In the secondary, it's IgG because it responds at the same basic rate, amount of time as IgM, but the response is phenomenal. Let's see. Oops, one last slide. We'd be remiss in not mentioning some of the control of the immune response. We talked about before kind of the activation of immunity, and we're not going to spend a lot of time on it today because we'll do it in another chapter. We're going to be talking about kind of genetic control. But control of the immune response is a very, very complex process. We do have, though, in the production of immunoglobulins, which is all I'm going to talk about here today, really, is that genetic control of the MHC, the major histocompatibility complex, driving our ability to respond to an antigen. If we think about what makes something a good immunogen, what's one of the characteristics? Being able to be processed and presented by the MHC. So if we go back to our discussions about the MHC, we know that that's a genetic control point, individual to individual. So I may have an immune response to a certain kind of antigenic challenge, whereas someone else may not. It's also species to species. We see some species are good antibody producers, while others are not. The whole thing is rabbits produce high levels of antibodies to proteins, while mice do not. Ironically, we use mice to produce some of the antibodies that we use in the lab, or we did use them. Now we take clones of those antibodies that were produced. We don't have to keep killing off mice to get antibodies. But the genetic control really talks about who's a responder and who isn't. So if we talk about someone not being a responder to a vaccine, it's that genetic control. So remember that while the immune response itself, pardon me, we've seen how complex it is. We see how many variables go into it, and all of the different points of processing in controlling the immune response of self to non-self, and then in the actual response to something that's foreign. We maintain genetic control within species and within individuals within a species. We talk about that and how that can create antibody diversity, not antibiotic, antibody diversity. And that because each of us have our own genetic makeup, we all respond to antigen challenges in different ways and antibody production. Humans can make anywhere between 10 to the 6th and 10 to the 9th different types of specific antibody molecules. That's an incredible number, and they're all genetically coded. They're all genetically coded for us. We understand the importance of antibiotic diversity. If we all were non-responders to a certain kind of pathogen, we'd all be dead. So we need to have antibiotic diversity, and that's an important feature of a host, a class of hosts. How did we get to the point that we, understanding what makes up this antibody diversity among a class of vertebrates? There's lots of hypotheses. I think they've kind of, landed where they landed, but it started with a guy named Ehrlich, who postulated that some cells had receptors for the antigen before the contact ever happened. Another gentleman, two gentlemen actually kind of simultaneously found that and proposed that, let's look at a typo, proposed that lymphs are genetically predisposed to produce, a certain class of antibody, and that the antigens then go select those cells capable of responding to it. Seems counterintuitive to me, but it was the hypothesis, and that lymphs proliferate and produce antibody in response, and that's called the clonal selection hypothesis. Now I think we know that there's genetic coding for immunoglobulin genes in cells, and it's that gene rearrangement that we've talked about in a couple of contexts between T cell and B cell maturation, that genetic rearrangement and the subsequent transcription and translation for synthesis of immunoglobulins that creates really antibody. I'm going to keep calling it antibody diversity. So I think we know now that gene coding plays an important part in this process of creating an antibody diverse species. I'm not going to go through the summary. You can look at that, but I am going to flip ahead really quickly to, there's some study guides. I want you each for a homework assignment, and if you look in the portal, it will have a deadline that is out there for you. I want you each to make an immunoglobulin table. It can look something like this. This would be me because, I'm in a chart form. And there is a chart in the book, but I want you to make this one that works for you. It's really important that you can classify each of the 5 classes of immunoglobulins, the IgG, IgD, IgA, IgE, and IgM, in a way that you can remember it. Identify each of the heavy chains, the classes associated with it, which is the most abundant, the least abundant, which has the longest and shortest half-life. How many subclasses are they? How are they structurally different? What are the functions of the immunoglobulins? What structure do they take? Do they cross the placenta? Do they bind complement? If there are subclasses, what are the differences? Which ones are secretory? What is secretory IgA? What's its function? How does IgM act in a secretory function, for example? What else can you put into this table for yourself that will make it work? You're going to make a copy of this and give me your copy. You're going to keep your original because you're going to want to study from that. And so this would be an example of something. You could certainly make this table however you like. You can draw it on a piece of paper in a notebook. You can put it on a chart, whatever you like that works for you. I've had people in the past color code it. I've had, people have all sorts of ways that they can use it as a study guide. And that's what I want you to do with this. So for homework on this chapter, I want you to make an immunoglobulin table, something like this. And you will also see a couple of questions in the portal or in the module with a deadline as well for homework. And there's our references. And that's the end of immunoglobulins and antibodies. I know it's a really big chapter. The original PowerPoint is loaded, and I'm going to upload this video as well. And you should come to me and send me an email or text message, or not a discussion post if you have any questions about anything on this chapter. I will talk to you soon. Thanks.